Thank you. So hi, everyone. I'm Olivia. I'm a lecturer at Massey University in New Zealand. And the research that I'm going to present today comes from my PhD project that I did at Massey University in collaboration with Plant and Food Research. So the goal of this research project was to investigate the molecular mechanisms behind the potato bruising in the tetraploid potato. And so the data set that we were using for this project comes with quite a complex multifactorial design. So the idea behind it is that two main parents, let's call them parent A and parent B, were used and crossed with a number of other parents. And what we are looking at for this project is actually the progeny from these parents that were subject to selection. So we have, as I was saying, a multifactorial design where we have full siblings in the population that we are looking at. We also have half siblings, and it even gets more complicated than that because some of the parents have also some relationships, parental relationships, etc. So for this project, we obtained genomic data using capture by sequencing using exon captures. So we had around 25,000 baits that were designed to target exons. And so the distribution of the baits across the chromosomes and across the genome reflect the exon density. We obtained genomic information for 178 samples. So most of them were progeny genotypes. And we also had information about some of the parents that were genotyped. And sometimes we also had several biological replicates for the parents' genotypes. The variant calling was originally done with freebase. And then later we tried to improve the genotype calling by using polyrad. And so in the end, after doing a filtering based on the quality of the reads, the minor allele frequency, the missingness, etc., we obtained a bit less than 500,000 biallelic SNPs in this data set. So it's quite a big data set in terms of the SNPs that we have. The phenotypic data that we have comes from a bruising experiment that was performed on a subset of the genotyped progeny data. So basically, the tubers were bruised by throwing a weight from a controlled height. And then after 24 hours, the bruising was recorded and then scored visually on a scale from 0 to 5. And so that was performed for several biological replicates. So the actual phenotype that we obtain is actually the mean of a bruising score, or bruising mean score, if you want. We also had other phenotypes recorded for this population. So we had things like dry matter, sugar content, maturity, etc. We also had other omics data sets, which was really interesting. So we had transcriptomics data that was obtained via RNA sequencing for 100 of the progeny samples. And it's also quite a large data set because after filtering, we have a bit more than 25,000 transcripts that were measured. And we also had metabolomics data that was obtained by LCMS for 122 of the progeny samples. The really good thing was we had for this metabolomics data set 2 biological replicates for each of the progeny genotypes. This was also quite a large data set with around 4,600 compounds that were measured. However, I have to note that for this metabolomics dataset, the main bottleneck is actually the compound identification, which is a really complicated step for untargeted metabolomics. So one of the first things that we did was to perform a genome-wide association study. And so for that, we used the GWAS poly package. Now, the thing is for this GWAS study, there is a really strong population structure because we have full siblings, half siblings, etc. So the first thing before performing the GWAS analysis, I looked at the population structure using both the structure algorithm and the DAPC algorithm. And the reason why I tried these 2 different algorithms rather than just using one of them is because they both give us a little slightly different information about the structure of the population. So I wanted to investigate which one was more appropriate to use to correlate for population structure in this case. For each trait of interest, we tested the different genetic models that are offered in the GWAS poly package, but what we wanted to test was how to properly correct for the impact of population structure. And so for that, we use different population models. So we started with a GWAS model that was the naive model. There was no kinship matrix, no correction for subpopulations, etc. We tried also a K model in which we included only the kinship matrix that was computed using the LOCO method. Then we tried including the subpopulation membership probabilities as covariates, so either coming from the structure algorithm or from the DAPC algorithm. So that's the Q models. And then we also tried the K plus Q models for each of the population structure algorithms. So that gives us a lot of models. So in total, we have 48 GWAS models for each trait. That's way too much. The reason why we did that was then we wanted to select for each of the phenotypes the best model that corrects as best as possible for the impact of population structure. So in order to do that, we computed the inflation factor of the GWAS scores for each of the GWAS models. We took the average over all the genetic models for a given trait, that's the columns here, and a population model, that's the rows here. And then we looked at which population model gives us the best average inflation factor. So ideally, if you correct well for the impact of population structure, you should expect an inflation factor that is close to 1 and hopefully above 1 to mean that we have some significant results. It was really interesting to see that the population structure had a really different impact on depending on the trait that we were looking at. Some of the traits were obviously impacted by population structure, where you could see a large inflation of false positive with the naive models. Other traits, not so much. And what we noticed is oftentimes, so the K model, adding the kinship matrix was often required in order to get nice results. And in general, the structure algorithm gave us better results because it gives us more information about the admixture of the samples between different subpopulations. The next step was to detect significant QTL. So we used the effective number of markers, which is a Bonferroni type correction, to compute the significance threshold and hopefully detect significant QTLs. What you can see in the figure here is the number of significant markers or of markers with significant scores for the different phenotypes, which are the rows here, and for each of the genetic model, which are the columns. And you can see that mostly it's empty. So we had mostly no significant QTLs. And it's really not that surprising, given that the population that we're looking at is not a very nice association panel or diversity panel, but it's data that comes from a breeding program. So there's an impact of selection that plays a role. And also we have a relatively small number of samples because we have less than 200 samples. So it's really not that surprising that we didn't get hits, especially for the bruising score mean, which is the one that we are interested in particular. However, what I wanted to show in this talk is that it's not because we didn't have significant scores that we didn't have biologically interesting signal. And so what I went on to do is to look at the peaks in the GWAS scores. So if you think about the GWAS result as this Manhattan plots where hopefully you see a really nice peak, I wanted to see where these peaks were and if they had any biological relevance. So the plot that you're seeing here is a zoom on chromosome 8. And so the x-axis represents the position along this chromosome. And then the upper track shows you the position of markers in whose GWAS score were above a certain threshold. So it's not a significant threshold, but it's the markers that had the highest scores. And then in the below track, you can see the position of candidate genes that were highlighted by previous studies as being potentially associated with tuber bruising. So the results are not that amazing here. You can see that we have some peaks in the GWAS analysis close to this region where we have genes of interest. When we were playing with earlier iterations of the model, when we were using free-base genotype calling, we actually obtained a really nice GWAS peak, even though it was not significant, just around this region. So that's maybe something to investigate more. But the interest is that we still had peaks in the GWAS, even though they were not significant, close to regions that we suspect are associated with tuber bruising. So that was encouraging because even though we don't have anything really significant, we still have kind of clues about where we should go digging more, in which genomic region we should go investigate the origin of tuber bruising. We, of course, can do. So the plot that I showed, we can do that at the scale of the whole genome, and then that can hopefully help us detect some genomic regions that might be having an impact on potato bruising, even though, again, the scores are not significant. Now, what was really interesting about this data set is that we didn't have only genomics data. And so we used the other omics data sets to try and better understand or interpret the results that we were getting. So in particular, we used the transcriptomics data set and performed a differential expression analysis with the DSEG2 package. So in total, we found 58 genes that were differentially expressed between samples with a low bruising score and samples with a high bruising score. And again, this is interesting, but it gets more interesting when you combine the results of the transcriptomics differential expression analysis with the GWAS results. And so to the plot that I showed previously, we can actually add one more track here, which is in the middle, which shows the position of the genes that were found differentially expressed. And now you can see that it's really quite interesting. So we have peaks in the GWAS analysis, and then we have differentially expressed genes that are not too far. So again, it still spans a large genomic region, but it's encouraging, and it allows us to kind of pinpoint which genomic regions should be further investigated. And again, we can do the same thing at the scale of the whole genome. And so again, we have this region at the beginning of chromosome 11, where we can see some peaks in the GWAS, and we can also see some genes that are differentially expressed. So maybe there's something going on in this genomic region. Now, because we also had metabolomics data, we wanted to do some multiomics data integration to try and make full use of all these data sets. And so for that, we used the Mixomics package. So maybe if people are familiar with multiomics data integration, that's a very famous package to use. And so specifically the DIABLO algorithm, what it does is given different groups, phenotypic groups, so we have samples with low bruising, we have samples with high bruising, the algorithm will select features from the different data sets that are associated with these different groups, so that best discriminate the different phenotypic groups. So we selected markers of variants, transcripts, and metabolomic compounds that were associated with these different bruising score groups. And that was really interesting because it actually allowed us to detect transcript and compounds that were not found differentially expressed. So we would not have picked them if we were analyzing the omics dataset on their own, but they're actually the ones that were selected were actually relevant for tuber bruising. Now, the reason why I wanted to talk about that here today is also because when we did the multiomics data integration, we realized that actually if you just give the variants that you have to the integration tool, you also have a very strong influence of population structure on the results of the integration. And that's something that's interesting. Yeah. Oh, sorry. So that's something that's really interesting because when you're doing GWAS analysis, you're very aware, or we are very aware, that we need to correct for population structure, but that might not be the case when we're doing other types of analysis. And in particular, integration tools are not equipped to deal with this type of biases. So I found that was really interesting to highlight that in this data set. Very quickly, the last thing that we did was to use different causal inference methods to try and assess the causal relationships between the features that we selected in the previous step. And so that gives us kind of a map. There's still a lot of work to do to interpret this map, but basically you can see the relationships between different types of features, so between different markers that were selected, genes or molecular compounds, and hopefully that will give us a clearer view of the molecular mechanisms involved in potato tuber bruising. In conclusion, I hope that this showed you that genomics data that comes from a breeding program, even though it's not a really nice diversity panel, can still be useful when you perform GWAS analysis to locate at least genomic regions that could be associated with a trait of interest. But I found that actually, in this case, adding other types of omics data, like transcriptomics or metabolomics data, was really interesting and provided alternative way to interpret the results and to detect maybe potential causal genes or even just biological pathway that could be worth investigating further. So of course, this work, I did not did not do that on my own. And so I would like to give a big thank you to my supervisors for my PhD thesis at Plant and Food Research, so Samantha Baldwin and Susan Thompson, and also my supervisors from Massey University, Matthew Vince and Patrick Biggs, and also all the people at Plant and Food Research that were involved in the data collection, analysis, processing, and also who just helped me develop this analysis. And on that note, I would be happy, thank you for your attention, and I would be happy to answer any questions.